I'll try to talk a little bit about the way we think about the thalamus, its role in the kind of context of the rest of the brain, and from a perspective that has a little bit of human neuroimaging, a little bit of computational modeling, and a little bit of kind of really fun, gnarly, cellular neuroscience. So a very, sort of broad talk, but hopefully it'll spur some really interesting discussion. But rather than starting from the thalamus, I want to start from something way more abstract, which is kind of how do we figure out how systems work? How do we figure out how to link structures of function? How do we figure out how to understand a concept as complicated as something like cognition, attention, arousal, emotion? And years ago, although it's much maligned, it's not the perfect way by any stretch of the imagination to think about complex systems like this. David Ma had a really nice way of breaking down complex problems into a set of different levels that interact together. He called these three levels the computational level, the algorithmic level, and the implementation level. We can think about these really nicely through the analogy of a bird flying. And so you can think about the computation that the bird's flying is trying to solve is the flight, the sort of the raising against gravity of the bird's body and the conducting forward over physical space. You could think about the way that it implements that flying, the feathers and the muscles and the wings pushing against the air, the wings being able to flap using particular muscles of the upper girdle. And you can think about the algorithm as sort of where the rubber hits the road. How is it that the bird uses the feathers and the musculature and its particular bone structure to flap so as to fly? And you can think about different systems like a plane achieving similar computations to a bird flight, but with very, very different implementation and very different algorithm. So rather than using feathers and flapping them, the plane uses a particular rigid wing structure and fast velocity that then can create friction that's higher on one side of the wing than the other, which can then cause an upward thrust. And then that the algorithm there is the different movement of the air around the shape of the wing. So very different implementation algorithms for a similar computation. So why am I bringing this up? Is that most of neuroscience, at least at the systems level, really starts out at the computation level. So one of the really sort of famous ways that we start thinking about, the function that a brain is trying to solve, one of the more popular ways to think about is what's called free energy minimization. So the idea would be that there's a particular kind of landscape of particular options, and then you need to sort of move around that landscape in order to achieve, optimal energy efficiency, and hence, pass your genes onto the next generation. If you start from that computation, you can then think about, okay, what kinds of algorithms could give rise to that computation? If there's a computational space, what kind of algorithms could I imagine that would give rise to that? And then once you've done sort of convincing yourself that you have a really nice algorithm that's really well behaved, you have nice mathematics, you can then sort of hunt around for potential links to neuroanatomy at the end of it. You sort of start with the computation, figure out the algorithm, and then go down into the implementation level. And so this is a really, really kind of infamous and quite popular account of something like perception, how perception would work in the brain. It's known eponymously as the Raoul-Ballard model, but the kind of broader scope is predictive coding. And the idea here would be that perception, in the case of seeing a particular item in front of you, isn't so much just the way the camera would work. It's not just about taking the particular pixels that are hitting your retina and just sort of directly interpreting them. It's more about a prediction that you're making, and then the match between those predictions and the evidence that comes in. And then the idea is that if those predictions match really, really well, the brain doesn't have to do anything. But if the predictions don't match the inputs that are coming in, you send a little error up the chain, and that can help you kind of figure out what you got wrong. So this is a kind of like a typical algorithm that we think about for perception. The problem is that there's so many different ways that you can match that to that computation to neuroanatomy. So this is a really, really lovely paper actually that kind of takes this idea of computation down to algorithm and then tries to go and look in something like the cortical microcircuit. You got here little layer 2-3 supergranular excitatory and inhibitory neurons in the granular layer, then in the deep sort of subgranular, infragranular layers. But you know, one thing that's really missing from this whole idea is really anything but the cortex, right? You're missing the thalamus, you're missing the colliculus, you're missing the neuromodulatory arousal system. All these parts that are really crucially involved in an organism perceiving the world are missing from this model. And so it begs the question, you know, how is it that the system could actually, how could you actually match the particular function that we're talking about, perception, to just one subset of the system? You know, the classic kind of cartoon to give you the feel for, you know, where this can go awry is that if you're only looking at one part of the system, here a set of blind scientists all inspecting an elephant, they all think it's something different based on the particular area that they're looking. So, you know, the ear looks like a fan, the tail looks like a rope, you know, the trunk looks like a snake. And I think we often make this mistake in neuroscience. We often start with a particular computation, and then we go down looking at the complexity of the nervous system, and we try to match a function to an algorithm to a particular implementation. But that is kind of really difficult, if you think about it, for a few moments, to be lucky enough to capture the part of the elephant that somehow crucially shows you its entire function is very unlikely from that perspective. In part because the number of cell types in the brain is just outstandingly massive. I mean, if you look at a human brain, this is just a sort of subsample of the number of different types of interneurons in a part of the human cortex. I mean, it's just astounding how many different types of these there are. And I think, you know, to imagine that you stumbled on just the right one when you were looking that sort of somehow fit your algorithm, I think it's kind of a bit of a difficult aim. And another challenge is that, let's say you wanted to match your function to a particular subcircuit, the one I was just showing you before, is actually based on a particular part of the visual cortex here that's actually quite different than lots of the other parts of the cortex. So this is what we call granular cortex. It's really common in V1. It's also it's part of the sensory areas in the auditory cortex and other parts of the frontal lobe. But many other parts really look nothing like that at all. And so the question is, if that function is sort of somehow matched to one subsection of the cortex, what about the rest of it? How does that fit into that story? So what I want to convince you today, and as I said, as people that are interested in the thalamus, this may be something that already naturally emerges for you because of its topological role in the brain, is that another way to think about it, really popularized in popular culture by Yuri Bujaki with his beautiful book, The Brain from Inside Out, is to start with implementation, start with the sort of neurobiological details, start with, you know, how the actual brain is organized, then try to figure out what kinds of algorithms could that set of structures imbue, what kinds of ways would they interact, what kinds of computations might that allow, and then you can match that to the kind of complex adaptive behaviors that we're trying to understand. Now, this isn't to say this is a sort of knockdown approach that somehow works out of the box. But the benefit of this approach is that when you actually come up with a solution, if you can, that links across these scales, you've done it in the language of how the brain is actually put together. So what you're actually done is you've sort of taken that searching problem and you've turned it from computation down to searching an infinite haystack for a needle to finding a detail and then expanding out to figure out what that could do. So I think that latter one, there's a lot more control in our hands when we're trying to sort of figure out the rules of engagement. But it also comes with an inherent problem, which is that when you go looking down the microscope at the brain, as I said before, there's just this incredible dizzying complexity to the way that the nervous system is organized. So this is a famous map from Fellman and Van Essen from back in the 90s. Terrifyingly, when I was still in primary school, where they mapped in here in a macaque the different structural connections between identified architectonic areas of the ventral and temporal visual stream and the dorsal visual stream in a macaque. And it's essentially an all-to-all connectivity map. I mean, it looks a little bit more like spaghetti than anything. It's very hard to discern some kind of functional architecture from this. You can sort of convince yourself that certain places are sort of above one another or before one another, but it's really doesn't lend itself to a really convincing sort of functional interpretation. The other challenge that we face, other than the kind of dizzying complexity of the nervous system, is the fact that it's organized across so many different spatial and temporal scales. So just to give you a kind of feel for that, so here we've got an individual, neuron. We've got the endpoint of, let's say, a presynaptic and a postsynaptic neuron with a little cleft between them. But we have to remember that synapse is but one of many, many, many synapses on a particular neuron. This is a little layer 5B pyramidal neuron, one of my favorite neurons in the cerebral cortex, studded with these little synapses all across its apical and oblique and basal dendrites. Now, this cell itself is embedded within columns within the cerebral cortex. So, interconnected, lots of different specialist types of supragranular, granular, infragranular cells, all connected together in this particular columnar architecture. But that column is just one of many columns, right? There's columns that spread all the way across the cerebral cortex, and they themselves are connected in complicated ways, both to other cortical regions, to the thalamus, which we'll talk about today, to different neuromodulatory systems, to different subcortical systems. And all of that ends up being one tiny little cube on the kinds of imaging that we do on a human brain at the scale of the whole brain. And so we've got across this incredibly diverse set of, in this case, spatial scales, but also temporal scales, if we want to understand the system from the ground up, if we want to start with the cells and build it up into a system. And so what I would argue is, and I'm not alone here, is that we need to think of simplifying heuristic maps of the organization of the nervous system. We need to figure out some of the kind of governing principles that let us know, okay, at a very fuzzy resolution, what's connected to what? What are the main axes of variation? How do those axes of variation affect the kinds of modes of operating of the nervous system? And in doing so, maybe then we can start to kind of tease apart some of the different algorithmic and hence computational benefits of the organization. To anyone who hasn't read it, this is one of my favorite papers in neuroscience from Marcel Messelem, again, written back in the 90s. I'm probably betraying, you probably can tell what my music taste is now that you know that I like papers from the 90s. But so Messelem, I'm pretty convinced that Messelem has traveled back in time, like he's from the future and he's figured out a whole bunch of neuroscience and then traveled back to the late 90s and written a paper. Because this paper is so prescient and so clear in the systems level way of thinking that many of the fields in which I read papers are still struggling with concepts that he laid out back in the 90s. So the idea here is that there's a sort of topological order to the cerebral cortex that links the outside world, which would be what we call the sort of unimodal areas, like in the visual cortex, the somatic motor cortex, the auditory cortex, with the other end, which we call the limbic system, or the sort of transmodal system, which sits at the other kind of extreme in the anterior insula and parts of the orbitofrontal cortex. And then in between are these different channels of connections that essentially allow information, as it were, to flow between different ends of the system. But rather than thinking about it as going sort of front to back, it's really these different specialist systems through a kind of complex network towards these valuation systems at the other end. So this is, this abstracts out so much of the complexity of the cerebral cortex, right? It's not talking about specific cell types. It's not talking about layers, connections, but it's giving you a kind of overview of like how to think about the different ends of the cerebral cortex in a way that's really useful for thinking about its function. Another really lovely perspective that I love from Helen Barbas's group called the structural model kind of takes this one step further and thinks about the layers of the cerebral cortex and how they interrelate together. So it comes up with a set of heuristic rules that determine the likelihood that a particular cortical area will project to another one in a particular way. So you can imagine if you're in a granular area of the cortex, like V1, the likelihood that you'll send projections to the supragranular areas of a upstream area is much higher if that area is less granular than V1. And the vice versa is true. The less granular area you are, the more likely you are to project back down to a more granular area and hit supragranular and infragranular areas of that cortical layers. And so this is sort of shown quite nicely in a really dense form here in this paper by Klaus Hilgertag's group, where you can see the more granular areas on the outside and the more agranular areas on the inside, and these different kind of connection paths that are really the same as this map here, but now linking them up into particular parts of the brain that then you can imagine as sort of informational channels that will link it between these different circuits, so that if there was a connection here, it could kind of pass back down the network and maybe move sideways and then travel its way back in. And so, in other words, it's a simplification. Again, it's not covering all of the details of all the interneurons and all the different neuromodulatory receptors and things, but it's giving you a feel for the kinds of ways in which the nervous system is organized that might betray its function. So with that Marian and corticocentric introduction, I want to argue that starting from the thalamus is actually a really, really intuitive place to start thinking about the organization of the nervous system. It's a topologically central location. It's interconnected with most of the other major processing structures in the central nervous system: the cortex, the basal ganglia, the superior colliculus, the cerebellum, major parts of the brainstem and hypothalamus, the amygdala, the hippocampus. All of these areas that we think of as sort of these sort of separate functional structures are actually really interconnected with the thalamus. And I believe that if you focus on the way in which those systems interact with one another via the thalamic intermediaries, a lot of that newfound perspective, that thalamocentric perspective, can really betray the functions that that system might be capable of. So in other words, instead of starting with the cortex, we start with the thalamus. The ability to build from implementation through to algorithm to computation, I think is a clearer path. So, I'm preaching to the choir here, but we all know that the thalamus is this fascinating structure with this really amazing psychoarchitectonic diversity all packed down into a really tight set of little nuclei in the diencephalon, all with these very interesting different connectional topologies. I remember early on when I was learning about neuroanatomy back in medical school, putting the thalamus firmly in the too hard basket, right? Rather than, it's very hard to sit down and learn rote exactly which areas project to which, and how to differentiate the pulvinar from the lateral dorsal nucleus, and the VL from the VA, and anterior from medial dorsal, and all these different areas they look really similar. They're in a very similar part of the brain, but they have very different types of connections to the rest of the nervous system. And so what I want to argue today and sort of talk a little bit about is kind of how to think about, or how I like to think about some of these systems embedded within the whole system. So here I've drawn a little kind of heuristic map of the different ways in which structures like the basal ganglia and the cerebellum, these two major subcortical structures that are really crucial for adaptive behavior, and the way that they interact with the ventral anterior and the ventral lateral thalamus. So a group in Japan, Kuramoto and colleagues, had this really lovely study they did in rodents, where they actually went in and they identified neurons in the ventral anterior and the ventral lateral thalamus in these rodents. And then they could figure out, according to the type of neuron they were, whether or not they were expressing a different type of a calcium binding protein. In this case, calbindin for the dark brown cells and parvalbumin for the light brown cells. And then they could also figure out, okay, if it's a calbindin cell, is it likely to be receiving GABAergic or glutamatergic inputs? And if they were glutamatergic inputs, were they GLUT1 or GLUT2? Now, the reason that detail is important is that GLUT1 and GLUT2 are different markers for whether or not the cortex or the subcortex are projecting and innervating that particular area. And it turned out that there was a really, really strong correspondence between the regions that were calbindin staining and GABAergic inputs, which in the ventral anterior thalamus are almost almost exclusively from the globus pallidus internus, and the parvalbumin staining neurons in the ventral lateral thalamus, and GLUT2 inputs, which are predominantly coming from the deep cerebellar nuclei. And this was a really key paper for my thinking when I came across it. I think it betrays a really, really crucial organizing principle. Now, like all things in biology, it's not a perfect one-to-one. There are definitely deep cerebellar nuclei that project to some of the matrix-like regions that are calbindin staining, and vice versa. But this is sort of a general rule for how these two systems interact with the thalamus. But the crucial thing is that they actually have extremely different projection topologies afterwards. So the basal ganglia projects to these cells, the calbindin staining cells that actually diffusely project up to the cerebral cortex and often cross psychoarchitectonic divisions and then also innervate the striatum, whereas the core type cells with parvalbumin staining typically project up and really precisely innervate the granular layers of the cortex. So we'll come back to these in a little bit. Overall, I think these kinds of connectional topologies are really portraying what we would call something like a functional primitive. The thalamus is really fascinating in the fact that it can support a diversity of these different types of primitives. So you can imagine that a particular thalamic region, if it is innervating a cortical area and then receives an input back from that same cortical area, let's say from layer 6 or layer 5 in the cortex, it can actually boost the activity of that particular cortical neuron and inhibit via the reticular nucleus a number of competitive neurons that might otherwise like to be the most active in the circuit. And so otherwise it can kind of boost the signal to noise in a particular circuit. Similarly, if a cortical, if a thalamic area is actually supporting, this particular nucleus is supporting either two different areas, or in the fascinating multi-specific nuclei in the thalamus, which Francisco Klaske talks about, you can actually have particular neurons that come up and their axon actually splits. And one of the axons goes off and projects in a diffuse way, and another one projects in a targeted way to completely different cortical areas. And so you can imagine now that that particular one thalamic nucleus, if it's active, can now essentially bring online coordination across distributed parts of the nervous system. If we scale that up, we can think about topological features like different thalamic nuclei supporting, say, a visual circuit, while there's also a separate one supporting, let's say, a memory-based circuit. And so the thalamus has the capacity to essentially keep different systems online at the same time, which I think is a really crucial feature of cognitive function. And the thalamus can also promote variability in that it can essentially allow a system to become more active and express more of its dynamic range, again, through boosting it from the bottom up. And so working with some collaborators from a few different groups, we're working on a review paper that essentially tries to kind of use this vantage point to think about a lot of different ways in which the thalamus interacts with the rest of the nervous system. But I think it's worth pausing and really thinking just about how diverse a set of functional primitives this really is. The difference between promoting the signal to noise activity of a particular region, but also its variability, and also coordinating its ability to couple, but also segregate from the rest of the network. It's really fascinating that a single structure, at least the way we think about it as a single structure, is capable of supporting such different functions with the same kind of an architecture. So this is a little graph here of some of those different connectivity principles that I was mentioning before. So the typical way we think about these parvalbumin sending neurons as a sort of core-like, they project in to the sort of granular areas and diffusely sort of innervate this local area of the cortex, versus these more matrix-like diffusely projecting systems that also then project into the striatum and parts of the central amygdala. And then there's also this other type, the intralaminar type, which really, predominantly subcortically projecting, but also have some projections to the deep cortex. So there's all these sort of dizzying different corticothalamic signatures in terms of the projection topologies. But one of the ways to sort of simplify this, which of course is a simplification, but I really like this idea, is from Ted Jones. And the idea is that you can sort of think about the sort of diversity of all these different types of connection principles along this kind of extreme of gradients. The idea is that at one end, you've got the really targeted type in green, the core type nuclei that project into the granular layers of cortex, and these have staining for parvalbumin. On the other hand, you have the matrix type, which project up rather diffusely and hit both the supragranular and the infragranular layers of the cortex, and often across different functional boundaries. Now, it's quite fascinating that these have different calcium binding proteins, because they perform exactly the same function in the cell, which is essentially just to kind of mop up calcium and make sure that you don't turn free calcium into some kind of awful precipitate with all the phosphate that's lying around. But it just so happens that through some accident of development, they're expressed in different ways on these different populations of neurons. And so one of the things we wanted to figure out was, can we take some estimate of their expression in the thalamus and figure out whether or not the different patterns of activity in the blood flow of the thalamus relate to the way that the rest of the system is organized? And so Eli Muller, a postdoc in my lab, essentially took gene expression data from the thalamus, from the Allen Human Brain Atlas, for both calbindin and parvalbumin genes, He essentially then extracted time series, voxel-wise time series of the dynamics during the resting state in a 7T data set from all of the different voxels within the thalamus, around 2000. And then essentially did the same thing just for every single voxel in the cerebral cortex. So essentially, rather than caring about the Calbine and Valbumin setting here, we're just trying to see what the kind of axes of variation are in the cerebral cortex. And then what he wanted to do was to sort of look for temporal relationships, linear temporal relationships, zero lag correlation between the populations with heavy Calbine staining, which we would kind of think of as reflective of the matrix population, and the core the Valbumin staining, which is reflective of the core. And essentially what he found was that there were really striking differences in the correlations between the core population and the cerebral cortex and the matrix population and the cerebral cortex. Again, estimated really, really roughly from the gene expression pattern. The matrix population was much more correlated with areas that we would typically call the default mode network, so in the sort of orbitofrontal cortex and posterior cingulate, as well as parts of the anterior insular and the frontoparietal cortex. Whereas the core population was much more correlated with parts of the visual system and the somatomotor system and the auditory system. For those who are interested in the resting state literature, there's a really strong correlation, positive correlation between the maps that we saw, the difference between them. So if we just take matrix minus core, we see a really strong relationship with the primary gradient. The idea here is that this is very much like what Marcel Messelin was talking about back in his paper in the late 90s, that this really simple system that differentiates sensory regions connected to the world and regions that are connected more to valuation. Much more complicated nonlinear relationships with the other gradients, sort of main axes of differentiation in the resting state, which you can sit and ponder over if that's something you're interested in. Another thing that we were able to do in the study was to essentially show that the areas that were correlated with the matrix actually had a much longer time scale than the areas that were correlated with the core. So this is calculating two different measures. One's called the Hurst exponent, one's called the M factor. Both of them look at the kind of time scale intrinsically to the bold signal, and you can essentially see a positive correlation between the matrix components and these data. And you can also then do fun things like look at what we call dynamic connectivity. So here what you do is you break the data up into little windows, and then you look to see how variable the patterns of correlation are in those little windows. And essentially what we found was that the areas with much more dynamic correlation patterns, the ones that changed a lot over time and switched their allegiance, so they went from connecting to A to B to C to D, were more likely to be associated with the matrix. Again, consistent with this idea of this sort of diffuse populations that could sort of change the variability of the coupling of these systems over time. So that's an empirical study to try to get a feel for kind of how this is working in the sense of the kind of more in that sort of mechanistic algorithmic sense that I was talking about at the start. Eli turned to a neural mass model. So Eli has a background in physics and is really interested in taking concepts from physics like dynamical systems and criticality and bifurcations and bringing them into contact with the complexity of the nervous system. And so whenever you do that, you have to make simplifying assumptions. And so in this case, what we do is we pretend that this little circuit here on the left that has an excitotory inhibitory population in the cortex and the same in the thalamus, which are then interconnected into a little loop. We assume that this is sort of like a kind of a little operating mode for a local area of corticothalamic loop. And then what we can do is we can wire them together into a really simple model. Eli used what's called a toroid, although I like to call it a donut. And the benefit of toroids for physicists is that there's no boundaries anywhere. There's no kind of cliff that the system could drop off of. And so every different part of the network can kind of interact with something else without having any edge cases. And then what Eli was able to do was, in a really, really simple model, was essentially just have a parameter that he could turn back and forth that essentially interconnected the whole population or made all the populations fire on their own. So it's a really, really simple population model, but you could think about this blue diffuse term as sort of representing what would happen if we turned the matrix thalamus on and we turned it off. And essentially what he found, I won't belabor the physics too much, but it's really informative because it turns out that there's actually a really tight analogy between what this parameter does in the model and temperature as you heated up a glass of water. So if you imagine you have a little glass of water that you apply like a flame to, what's going to start to happen is there's going to start to be more kinetic energy amongst all the water molecules. They're going to start to sort of bounce around each other more and more. But you also have the effect of actually spreading out the diversity of all these different states. And the way you can think about that is that if you start to heat up the water molecules, they all start to move a little bit more. But there'll just so happen at any one time to be one water molecule that thinks it's a little bit hotter than it actually is. It just so happens to get bounced into by 4 or 5 other different water molecules in a given window. Whereas there'll be another molecule that thinks it's a little bit cooler than it is. Because those molecules are over here bouncing into this guy, they don't bounce into this one. So essentially what you've done is you've sort of, by turning up the temperature, you've increased the diversity of different states that are possible. In the language of physics, what we're talking about is taking a system that's subcritical and turning up the temperature, i.e. turning up its control parameter, so that the system can actually cross a bifurcation and become supercritical. And what we think is actually really important is that the brain doesn't actually want to become fully supercritical, because that would actually be a little bit like an epileptic state. You'd actually lose the ability to process information. So we think that one of the things that the diffuse projecting thalamus, like the matrix thalamus, can do is actually promote this kind of what we call a spread of states, or a quasi-critical state, that allows both some cold areas and some hot areas of the brain to actually kind of live together in the same space. And so a way to think about that is, as you turn up this diffuse parameter from no matrix here on the left to high matrix on the right, what you actually do is you drag the system through this zone of quasi-criticality that actually has lots of information processing benefits. And one of the cool things that we can do is we can go back and look in the model, and we can say, well, what was it about the subcritical brain? It was different than the quasi-critical and the saturated brain. And it turns out that the main thing that we were changing is the balance between the reticular nucleus and the relay nuclei in our model. As we turned up the diffuse gain, what we were essentially doing was giving the cortex enough energy that it could overcome the inhibition of the reticular nucleus and now allow this sensory relay nucleus to essentially come back and activate the excitatory nucleus. So you sort of think about it a little bit like taking the reticular lid off the thalamus. If we do that too much, the whole system explodes and the sort of pot, boils over. And you essentially, you have this problem of essentially like evaporating your brain. But right in that mid zone, it's a little bit like really is like boiling a dish of water on your stovetop. You can see now that at that right temperature, different formations of water can evaporate, but then quickly cool back down and allow a new state to form. And we think that this feature, this analogy to water, is actually a really crucial sort of a pointer towards what we think the algorithms that might be supported by systems like this diffusely projecting matrix thalamus might be. All right. Another cool thing you can do with this modeling is try to recreate really fascinating results from the literature. So this is a paper that came out a couple of years ago from Yuri Solman's group. And essentially what they did is they had a couple of macaque monkeys, which they anesthetized with urethane gas. And then they were actually able to go in and stimulate the central-lateral thalamus, which is sort of in that intralaminar or matrix-like category, really in that kind of Calbinen end of the distribution, and stimulating that monkey's brain in that precise location, but not in the VPL right next door, which is more core-like, was sufficient to actually wake that monkey up from anesthesia. So the monkey's sitting there anesthetized in the surgery. They stimulate the central-lateral thalamus at particular frequencies, and the monkey wakes up and engages with the world. And if they do it in the VPL, it doesn't. So this is this fascinating switch between arousal states controlled in large part by this diffusely projecting thalamic system. So to be able to test this idea, Eli actually had to make his model a good deal more sophisticated. So essentially what we were trying to do now is take the model we had before, which was really just like everything on the left here, and now bake in a whole new population of these matrix nuclei with more diffuse projections. This requires a bunch of engineering, but what we can then do is we can bake that into a kind of a realistic, connectome of a human brain using diffusion-weighted connectome, which sort of estimates the white matter connections between all different cortical hubs. And then if we take empirical data, we can sort of essentially simulate bold data and get a really nice match to the data, which is really consistent with kind of leading models, which are really only and purely cortico-cortical, right? So it's very uncommon for people to model human brain imaging data and actually think about the influence of the thalamus on the rest of the system. They really sort of stay perched at that cortico-cortical level. Eli can then anesthetize the model essentially by turning up the extent to which GABA, the inhibitory influences, act on the network. That's essentially like turning up the inhibitory parts of the reticular nucleus and the inhibitory population in the cortex, and essentially can anesthetize these brains and then match kind of the types of signatures you would see in typical neuroimaging studies of anesthetized states. But then importantly, what Eli can then do is he can stimulate a matrix location or an area that has much less matrix-like input, right? Because we have a diversity of these different nuclei. And he can essentially recover all of these different types of signatures that people see across the literature. So in the Salmon data, they use this coherence in the alpha band. There's another paper in Science Advances just recently where they showed that stimulating the thalamus can actually increase the diversity of brain states. But there's all these other types of measures: integration, the time scale, the Kolmogorov complexity of the network, the susceptibility, and in each case, stimulating a matrix-like part of Eli's model is sufficient to actually match really, really well the waking data signatures. But if you do it to a low matrix compartment, you really don't match it at all. It really looks just like the anesthetized brain, which I think is really cool. Rather than just matching the data, what you can then do is you can actually figure out why it did that. And it's again, it gets into the realms of physics, but one of the really cool things that you can then do is you can say, well, if that worked, what was it about the model that allowed the system to have all these different signatures? And it turns out that in the wake brain, and when you stimulate the high matrix regions, what you're essentially doing is you're putting the brain into a particular mode such that spatiotemporal waves can actually interact with one another. You're causing what we call parallel activation of the system. You're bringing all of those systems online, but you're heating the water up just enough so they can all bubble around and interact with one another. Whereas if you have an anesthetized brain and you stimulate a low matrix area, what you essentially do is you cause these little waves that sort of just track each other around over time. They don't have that same kind of everyone online at the same time feeling. It's more like a ball that rolls down a hill and just continues rolling. So we really think that this kind of online together mode of parallel activation is a really crucial way to think about the benefits that the matrix system is actually providing for the brain. And then another thing you can do is essentially test these really cool ideas about information flow. So you can take really well validated measures of information storage and information transfer. You can calculate them on the kind of regional activity and connectivity between areas, and essentially show that in the anesthetized brain, what you essentially have is a sort of really high storage and low transfer. But then when you stimulate the matrix thalamic nuclei, what you get is you get a decrease in the storage and an increase in the transfer around the brain. What that essentially means is that the individual neural regions stop caring so much about what they're doing and care more about what the rest of the network is doing and let it influence them. Another way you can think about that is that the energy landscape of the system becomes a little bit flatter in the waking brain. And when you stimulate the anesthetized brain with that high matrix stimulation, what you're essentially doing is recovering a flattening of the landscape. For those that aren't initiated, this is a way of thinking about how the kind of brain state evolves over time. Is it changing in a really surprising way, or is it really stuck in a particular state? And the matrix thalamic nuclei is really buying you this diversity of states, this flexibility to ongoing information processing challenges. So just to reinforce that idea, here's like a little cartoon. So you can imagine a current brain state sitting on top of a little energy hill. And the brain state is going to evolve by following gravity down into a particular state. So you can imagine listening and following my slide is a little bit like a ball following gravity, watching this particular image. But imagine now someone knocks on your door and wants to get your attention. That's a little bit like flattening out your landscape, which is what we think the matrix thalamus is doing. It's buying you that local flattening, which then lets the system reconfigure so that a new attractor can form and you can move into that new brain state. So you can see that, these really abstract models in physics can have analogies to things like water to a ball rolling around a landscape, but they actually have a really deep mathematical basis that then lets us to sort of build the idea of implementation moving to algorithm so that we can understand what the computational benefit of that might be. All right, I'm going to skip through this one really quick because I want to talk a little bit about the systems level context and stuff that I think is maybe super cool for this group. So I mentioned before that I think that these systems, the basal ganglia and the cerebellum, are really crucial to think about because they have this diversity of different connections to the ventral anterior, the ventral lateral thalamus, but they specifically target, the globus pallidus targets the matrix thalamus. and the deep cerebellar nuclei target the core thalamus. And I think that this connectivity principle is actually really crucial for thinking about the benefits of having a basal ganglia-like mode or a cerebellum-like mode of processing. So here's that paper I was mentioning before from Kuramoto and colleagues back in 2009, where they went through and tracked GAD67, which is a measure of GABA, and that GLUT2, which is a measure of subcortical input, and then they could actually figure out that the GAD67 was much more likely to input onto a calbinin staining matrix like nucleus, and the GLUT2 staining inputs were coming onto the parvalbumin staining core like nuclei that are coming into those granular and areas of the cortex. So what might the implications for this be, right? These are such different types of circuits, cerebellum driving this targeted feed-forward projection to granular, and the basal ganglia gating this more diffuse projection. So in the case of the cerebellum, its connectivity is actually really well known, but it's incredibly complicated. It's one of those areas that you kind of have to read about again and again and again. You got to get a feel for what the sort of circuit topology is. But this is a really beautiful paper for those that are interested, Egidio D'Angelo's group. And this figure in particular, I think, really helped me to understand what the cerebellum's connectivity was early on. The idea is that the cerebellum is extremely high dimensional, right? Almost 50% in some estimates of the cells in the human brain, in the adult human brain, live in the cerebellum. And the vast majority actually live in this population called granule cells in the cerebellar cortex. And they have this really funny projections that end up looking like telephone wires that run up here. They're actually called parallel fibers that run kind of parallel to one another. And this same architecture is just repeated again and again and again and again, all the way across the cerebellum. Here they make contact with these Purkinje cells, which have this really funny, again, they're kind of orthogonal to these parallel fibers, and they're organized in these sheets. And the dendrites here of these cells connect into these wires, and they project down. The main output of the cerebellum is this deep cerebellar nuclei. But the really crucial thing is where this input is coming from, these mossy fibers. And it turns out that it's coming from the pontine nuclei. So the main output of the cerebral cortex, these layer 5 pyramidal neurons, every time any one of them burst fires, or it sends this projection down here that's heading down into the spinal cord in most cases, if you're in the motor cortex, but also down into the colliculus and parts of the thalamus and such. But a large majority of these also send a little innervation off into the pontine nuclei. Now, the pontine nuclei are this huge population of cells in that big bulge in the pons that you see if you look at a human brain in the sagittal section. Now, those inputs then project into that granule cell population. So that means every time you make an action or a movement, you're sending a little copy of that input into the cerebellum, which then expands like mad in its huge diversity of granule cell populations into this massive diversity of cells, all the different ways that that action could be translated. And you learn over time through matching in particular complicated ways, which particular outputs you want to project to, which then project back to the cortex via the thalamus in a really, really precise way. So in other words, what you've got is an action that then suggests an anticipated consequence of that action. So how might that play out? So imagine we've got this action here, we send a projection down, it goes into the cerebellum, we have some machination over what we ought to do next. What that's like is essentially suggesting the next attractor that could form. And if we can learn particular sequences of those, it'll be a little bit like that, those attractors forming in such a sequence that now we don't have to think about what happens next. So I think this is actually a really great explanation for what happens when you learn a particular skill so that it becomes automatic. Think about the first time you ever started driving a car. It was really overwhelming. You've got the steering wheel, all these different switches, there's mirrors everywhere you look, there's cars zinging past you, different pedals to push. It's overwhelming. But as you become more comfortable with that system, you learn how when you pull on the steering wheel, it moves this way and that. You push just so on the pedal, it will respond in the way you'd like it to. You learn to anticipate the way that that particular action will lead, what kind of consequences that will lead to. You essentially can just play the system in this mode where a particular trigger can cause the system to play out the particular sequence. So there's lots of lovely evidence for this that I review in the paper, but if you're interested, I think this is a really, really fascinating way to think about the way that the cortex via the cerebellum to the thalamus back to the cortex can actually project a particular computational benefit. And so it reminds me of this little gentleman, practicing his hockey sticking while he's balancing with the wind flowing, and he's able to do this while keeping his eyes up, on the display in front of him. So it's our ability to anticipate the sort of sensory consequences of our actions, such that we don't have to focus on them. I think there's a lot of really lovely evidence that the thalamus plays a really crucial role in communicating that information. So if the cerebellum is expanding the dimensionality, what's the basal ganglia doing? It actually looks like it collapses the dimensionality. So the basal ganglia has actually got far less cells than the cerebellum. In the same way that the granule cell population kind of blows out and is half the cells in the brain, the basal ganglia has far less cells than the cerebral cortex or the populations that project to it. And the striatum then projects down into the pallidum, globus pallidus, externus, and internus. They've got even less cells, which then project onto the thalamus. So you've got this real collapsing topology rather than the expanding topology of the cerebellum. But then it becomes even more fascinating that it actually projects back to diffusely projecting systems that then kind of re-innovate multiple different areas in a kind of very different topology. To get a feel for why this might be the case, there's a really lovely paper, if people are interested, by Josh Burke that talks about the basal ganglia and dopamine. And the idea is that dopamine is really good for both kind of rewarding and reinforcing previous states, but also helping us choose what we'd like to do next, what our potential actions are. And so you can think about things that sort of further back in time are very hard to reinforce. But if you get a dopamine burst, what you can essentially do is you can create plastic change that ensures that you will do that thing again next time. But importantly, it also invigorates your action. So you kind of move towards that now. So it's got this kind of like double-edged sword. One of them is facing forward, helping you work out what you want to do now. The other one's this sort of facing backwards. It's saying, I want to make sure that if this happens again, I do this again in this context. So if we put this back into our little model of the nervous system, now imagine we've got two little options here, A and B, that are projecting down into the basal ganglia. And now it's controlling, via this little collapse of dimensionality, it's controlling which circuits get reinvigorated up at the top level by these matrix thalamic nuclei. So imagine, you try something, you try option A and it's really rewarding, but then all of a sudden, you're really hungry, and then you smell some food coming, in the distance, someone's cooking something beautiful in your kitchen. Your dopaminergic system kicks on and says, now it's time to go, stop listening to this talk and go get some food. So what you'd really like to do is then you'd like to invigorate the system. You'd like to sort of increase the diversity of possible actions and flatten your landscape so that the ball can roll into that attractor of going to get some food. If, on the other hand, you're enjoying this talk, maybe you ignore the smell of food and you stay located in the current state and you don't move, right? You want to have the ability to stick around as well if you'd like. And so the idea here is that the basal ganglia is essentially buying you this ability to do what we'd call the swerve. It's giving you the capacity to invigorate possibilities for action that are different than the one you're in, but in a way that the system is able to still maintain the current state if it's still valued. So in other words, the basal ganglia can help you kind of ensure that the message gets communicated, whereas the cerebellum can help you carry the message and ensure that the message gets pushed through to its functional conclusion. I'm realizing that I'm running out of time, so I won't belabor the point, but I have a paper coming out soon that sort of thinks about the same kinds of an architecture, but more in the more posterior parts of the thalamus, specifically in the pulvinar and medial dorsal nucleus as well, in terms of how they interact with the superior colliculus. And the really fascinating thing is that the superior colliculus, this really crucial structure for perception, has different layers, and the different layers actually project to different parts of the thalamus. So the superficial superior colliculus projects to the inferior pulvinar, which has a predominantly matrix-like population, which then goes up and innervates large parts of the parietal and temporal lobe. And it really looks like it's playing a role in kind of augmenting the kind of coherent information amongst the system in that parietal lobe, so that if you have a particular item that comes into your visual display and it suggests a particular object, let's say, or maybe the location of a particular object, if the colliculus also gets that message, it can project up to the pulvinar and say, okay, whatever object you're seeing here, I'd like it to be able to project back and reinforce the particular information that's coming in, and they can reinforce one another. In fact, if you inhibit this part of the superior colliculus, people are essentially functionally blind in that location in their visual space. In contrast, the deep parts of the superior colliculus, which are really involved in saccadic eye movements, actually project back to the medial dorsal nucleus, and specifically to the parvocellular population, which project on, the multiforms population, which project onto the frontal eye fields. And so that you could think about this as a little bit like an efference copy. And so then you could start to imagine, well, how is that playing out? Where I'm looking, how is that playing out with what I value, right? What the basal ganglia is gating via these circuits in the medial dorsal nucleus to areas like the anterior cingulate, and what the cerebellum suggests ought to come next via these areas in the granular cortex, like the dorsolateral prefrontal cortex. And this isn't to suggest that this circuit is, all there is, but I think really the idea is that if you start with implementation, you can start to build that up into algorithms that then can give rise to that kind of computational level understanding of how a nervous system could solve the problems it wants to solve within the confines of the particular anatomy that it has to play with. So with that, I'll conclude 1 minute to 10 o'clock to say, man, the thalamus is beautiful. And it's so much fun to think about the rest of the nervous system from its perspective. If we sit in the thalamus and think about how the cortex is interacting with the thalamus, how it interacts with the basal ganglia, the cerebellum, the colliculus, and what kinds of functional modes the different types of thalamic nuclei can buy the different parts of the cerebral cortex in terms of diversity of states, or binding together connectivity or boosting activity. I think it's a really beautiful place to start. So to bring it back to our original way of thinking, rather than starting from the top and thinking about computation, what I really want to encourage is thinking about robust neurobiological features and mechanisms, the kinds of idiosyncratic algorithms like the kind of boiling water idea and its connections to physics, how that might relate to complex adaptive behavior. But really what I'd love to do is then be able to come back down and say, well, that explains some part of complex adaptive behavior, but what else can we do with our brains and minds that we can't explain in that simplified model? What else should we be looking for? What kinds of features should we be looking for in the nervous system? And ultimately what we really want is a dialogue between these different levels so that we can actually come up with good explanations for how the brain works. So with that, I'll say thank you. I think I showed you a lot of Eli Muller's work today. He's a really brilliant postdoc in my lab, but really it's a massive collaborative effort across the group and with all my collaborators. So thank you very much.